Hey there! Are you here to learn about DNA replication? Yes? Amazing! Then let's begin our deep dive into the wonderful world of DNA. Hold on, because it's going to be a wild ride. First off, let's get into what DNA is composed of. Nucleotides. What are those, you ask? Well, monomers, of course. Oh, you don't know what a monomer is? Essentially, they're building blocks for macromolecules. A bunch of monomers that can be put together to form a polymer through the process of polymerization. This is the chemical reaction that results in a bunch of monomers being chemically bonded together to form a polymer. In DNA's case, it's a condensation reaction where the energy of the removal of two phosphates, pyrophosphate, from the incoming nucleotide is used to bind the monomers together. We already know that DNA is composed of nucleotides, but what are these nucleotides? Well, they're molecules that are made up of a nitrogenous base, a phosphate group, and a sugar. There are 4 different nitrogenous bases in DNA that are super important in its structure: adenine, guanine, thymine, and cytosine, but we'll just call them A, G, C, and T from now on. These bases comprise the base pairs that are held together by hydrogen bonds. These base pairs only form A to T or G to C, meaning that these are the perfect pairs to ensure that the structure of DNA is up to par. Now, the phosphate group is very important for directionality and the polymerization of DNA. The sugar in DNA is called deoxyribose and is coupled with the phosphate group. These make the backbone of the DNA molecule. That essentially means that it's the support for the DNA as it forms a helix. A helix? What's that? It's the actual structure of the DNA molecule, composed of nucleotides, which are composed of the bases, phosphate groups, and sugar. It looks like a pretty twisted ladder with the base pairs in the middle forming the rungs, while the phosphate and sugar form the beams on the outside. I'm pretty sure everyone has seen this helical image before. It's such an iconic picture, it even has an emoji. Now that we know a little bit more about the structure, should we now get into the interesting part, DNA replication? Yes? Then let's jump into it. So replication. Do you know anything about it? No? Well, that's a bit awkward. Don't worry though, let's learn together. So DNA replication is essentially how DNA is copied in order to be passed down to future generations. It involves the use of one strand of the original parent DNA, known as the template strand, in order to form a complementary daughter strand. In the end, 2 new strands are formed. There's also a whole bunch of enzymes that help catalyze the replication. Let's walk through the steps of in vivo replication, the replication that occurs inside the cell. Now, the first thing that needs to happen is replication fork formation. This is done through the help of these 3 enzymes: topoisomerase, DNA helicase, SSBPs, single-stranded binding proteins. First, topoisomerase 2 helps relax the supercoiled DNA in order to relieve the tension created by unwinding DNA. Hey, remember when we talked about how base pairs were held together by hydrogen bonds? Well, that's something that's going to need to be broken in order for replication to occur. This is done by the second enzyme, DNA helicase, which disrupts the hydrogen bonding between base pairs in order to separate the strands into a Y shape. This Y shape is known as the replication fork, and this is where replication will truly begin. Oh, we almost forgot the last enzyme for this step. SSBPs, single-stranded binding proteins. These enzymes coat the single-stranded DNA and bind to these regions in order to prevent the template strands from coming together. Pause. We need to take a quick moment to explain a word we touched on previously: directionality. Directionality in DNA refers to the end-to-end chemical orientation of a single strand of DNA. The deoxyribose, the sugar in DNA, is numbered 1 to 5 based on the carbon atoms in the structure. The numberings are referred to as 1', 2', 3', 4', and 5'. It's important to know the directionality of DNA because it can only be synthesized in a 5' to 3' direction, with the 3' end having a hydroxyl group, OH, and the 5' end having a phosphate group. Now that we have that done, we can finalize this step by touching on the replication fork. We know that the replication fork is where replication will begin, but in what way? Well, when we separated the DNA strands with the DNA helicase, we formed the basis for the leading and lagging strands in DNA. These strands are named based on their orientations because they determine how DNA will be synthesized on each strand. The leading strand is oriented in the 3' to 5' direction from the fork, while the lagging strand is oriented in the 5' to 3' direction. As we get further into DNA replication, we'll learn of the significance of these orientations and how DNA is synthesized differently on each. Now let's move on to the next step, primer binding. Here we'll introduce a new enzyme, DNA polymerase or RNA polymerase if you're fancy. This enzyme helps to synthesize primers on each 3' end of the separated DNA template strands. These primers are what will help initiate replication and are super important for the process. If we don't have these, then replication won't start. Okay, let's head to step 3, elongation. Now there's another enzyme that we need to introduce, DNA polymerase. This enzyme will create the new strands and is super important for replication. How this works is that DNA polymerase binds to the primers that have been replaced on the 3' ends of each template strand. There, it will add new base pairs that are complementary to the strand in a 5' to 3' direction. This is where the orientation of the two previous strands come to play. The leading strand is oriented in the 3' to 5' direction, meaning that the 3' end points towards the replication fork. You know what that means? No? Well, that's okay. I'll explain how awesome that is for this strand. It means that the DNA can be synthesized continuously. So as the DNA is unwind, it can synthesize a new daughter strand without any interruptions. This isn't the case for the lagging strand, because its orientation is 5' to 3', meaning the 5' end is pointed towards the replication fork. Now this is a problem because DNA can only be synthesized in the 5' to 3' direction. We can fix this by binding several primers to the strand in order to synthesize the DNA in fragments, called Okazaki fragments. This process of replication is described as discontinuous because it isn't synthesized in one uninterrupted strand. Since DNA is now being synthesized, we can head to the last step, termination. This is where another type of DNA polymerase comes to the strands and replaces all the RNA primers with DNA, as well as proofreading the DNA, along with the original polymerase, at the same time in order to catch any mistakes that may result in a mutation. DNA polymerase is super good at this. It can catch 99% of all mistakes. Pretty good, eh? For the lagging strand, DNA ligase, the last enzyme for now, catalyzes bond formation in order to join Okazaki fragments to ensure the strand is continuous DNA. Welp, that's the end of DNA replication. In vivo, at least. You thought we were done? Nope. Not yet. We have one more topic to discuss. Did you catch how I've been describing this replication as being in vivo? Well, that's because there's another type of replication called in vitro, and this takes place outside of the cell. This is done in a lab environment and has been coined PCR, which stands for polymerase chain reaction. PCR is considerably less complex with only 3 steps and 4 essential components. The 4 essential components are template DNA, DNA polymerase, all 4 deoxynucleoside triphosphate bases with the bases A, T, G, and C, and 2 types of primers. Template DNA consists of 1 molecule of double-stranded DNA which contains the regions to be amplified. Essentially, the amplified region contains the DNA of interest and what we want to replicate. DNA polymerase is an essential enzyme in PCR because it is used to replicate the DNA. All 4 DNTPs that contain the bases A, T, G, and C are used as building blocks for the synthesis of new DNA strands. And of course, the 2 primers, which are called oligonucleotides, are used to start the replication as DNA polymerase needs something to start from in order to synthesize the new DNA. Now that we have the components down, let's move on to the actual steps. The 3 steps are denaturation, annealing, and extension. The first step, denaturation, is where the paired complementary strands of nucleic acids are separated. This is done through heating the solution in a plastic tube to a temperature short of boiling, approximately 95 degrees Celsius, so that the individual DNA strands of the template denature as a result of the breaking of the hydrogen bonds between the bases. The next step is annealing, in which the primers bind to their complementary sequences of single-stranded DNA via base pairing. This is done just as the solution is cooled down, which is about 55 to 60 degrees. And the last step is extension. Extension occurs at 72 degrees Celsius, and is where the reaction is heated to the optimal temperature for DNA polymerase, and each primer is elongated by means of DNTPs. There you have it. That's PCR. Now that we've finished exploring like DNA replication in vivo and in vitro, what would you say is your favorite? Oh, you can't decide? Me neither. I think they both have their perks. Well, that's it. Nothing else. Hope you enjoyed learning about that. See you. Bye.